Hi, I'm Lyudmila Kovalchuk, and I'm the project manager for BAXERNA, which is an Horizon Europe-funded project to develop new mRNA vaccines against clinically problematic bacteria, such as the causative agent of tuberculosis, using innovative immunopeptidomic workflows and novel vaccine formulations. To do this, we've gathered a team of experts in all phases of the process, starting from immunopeptidomics and analysis, vaccine formulation, all the way through to preclinical testing, and finally, a clinical trial team.
Hi, I am Charles van der Henst, and I'm a group leader at the VIB Center for Structural Biology.
And hello, my name is Alexandra Mor, and I am postdoc in Charles' lab.
And we are experts in Acinetobacter baumannii bacteria, and our goal is to provide samples for immunopeptidomics.
And to do so, we need to infect cells, eukaryotic cells, and to provide samples for the BAXERNA project.
So hi, I'm Francis Impens. I'm the coordinator of the BAXERNA project, together with some colleagues at VIB and Ghent University. And our role in the project is, in the beginning of the pipeline, we will use our unique mass spectrometry technology to identify novel epitopes and antigens of the bacterial pathogens that we want to tackle with the vaccines.
I'm Lennart Martens from VIB and Ghent University. I lead the Compomics group, and my group is purely based on bioinformatic work. We use artificial intelligence tools to get the most out of high throughput data. Very specifically, within the context of the BAXERNA project, we are dealing with the mass spectrometry data that has been acquired on the immunopeptides, and we then use our advanced AI tools to turn this data into identified epitopes, and these epitopes will then form the basis of the future vaccine design.
Okay, we are Dr. Lorenzo Franceschini.
I'm Professor Karim Brakpot.
And we are from the Laboratory for Molecular and Cellular Therapy at the Vrije Universiteit Brussels in Brussels, Belgium.
Our lab has almost 40 years experience in mRNA vaccine technology and immunology, and we would like to apply this knowledge to help to develop innovative bacterial vaccines.
We have a team of postdoctoral researchers, PhD students and lab technicians that is very excited to work together in this interdisciplinary consortium where both academic institutes and industrial leaders are involved. And we are looking very much forward to show what we can do with mRNA and to leverage our knowledge to help generate these innovative vaccines.
Hello, I'm Ine Lenteker. I'm a principal investigator in the Ghent Research Group on Nanomedicines at Ghent University. And I'm thrilled to kick off the BAXERNA project that we are jointly guiding together with VIB. Together with my research team, we are very excited to bring our expertise on adjuvanted mRNA vaccines to the project. And we aim to contribute to the development of more effective and widely acceptable bacterial vaccines.
My name is Gerben Marsman. I'm a project manager at the Vaccine Formulation Institute. The Vaccine Formulation Institute was founded in Switzerland in 2012. Our mission is to provide clinically relevant adjuvants to vaccine developers worldwide on an open access and equitable basis. We perform research and preclinical development of adjuvanted vaccines and disseminate vaccine formulation know-how to the entire vaccine community. In BAXERNA project, we will develop analytical methods to quantify the lipid components of novel candidate mRNA vaccines and assess their chemical stability.
Hello, my name is Selena Pavu and I'm a project manager at Quantum Biosciences. Universals is a global life science company created in 2013 with a mission statement of making biologics accessible to all. We believe that for a drug to exist is not enough. To be able to change the world, it's really important that everyone has accessibility to biologics, and this is really our guiding principle, accessibility in terms of pricing and quality. Quantum Biosciences is an entity of Universals, and our mission statement is to remove barriers for mRNA-based vaccines and therapeutics. As part of the consortium, we are really looking forward to working with all of the partners to use our formulation platform and help make better vaccines in the future.
My name is Pieter-Jan van Bokstal. I'm project manager at Reavita. Reavita is a company which is specialized in development of a continuous and controlled freeze-drying system. Our role in the BAXERNA project is to improve the long-term stability of the developed LNPs. We will take up the role to develop the formulation, which will be freeze-dried, and the process optimization.
I'm Caroline de Mongel. I lead a lab in the Pasteur Institute of Paris in the immunology department. So we are a team of 12 people interested in how bacteria suppress the immune system to establish chronic infections. And in the BAXERNA project, we will be involved at 2 stages of the project. A very early stage where we will help identify new epitopes presented by human cells when infected by these pathogens. And we will also be involved at a later stage when hopefully new vaccine candidates will be generated. We'll be testing them in the mouse model and seeing if the vaccine confers any protection.
Hello, I'm Eric Muraille. I'm Research Director at the Scientific Research Fund, attached to the Free University of Brussels. My team has extensive expertise in studying the immune response against infectious agents. Within the BAXERNA project, our task will be to evaluate the protective capacity of RNA vaccine candidate against Acinetobacter baumannii in a lung infection model in mice.
Hi, my name is Karin Rory. I'm a professor in vaccine immunology at the Karolinska Institute in Stockholm, Sweden. My lab is working on understanding how different types of vaccine platforms interact with the immune system to induce good vaccine responses. My involvement in BAXERNA is to perform late-stage preclinical studies of selected vaccine candidates in this consortium to be able to prepare for them to go forward in clinical testing.
Hi, I'm Mihai Netea. I'm an internist and infectious diseases specialist at the Radboud University Medical Center in Nijmegen, the Netherlands. Our group has expertise in trying to understand the host defense against infections in general and the innate immune responses after vaccinations. In the BAXERNA trial, we will investigate whether the new vaccines that will be developed in the projects have also impact on innate immune responses and long-term effects on inflammatory responses in healthy individuals.
So my name is Karim Vermaelen. I'm MD, PhD and pulmonary physician here at Ghent University Hospital. I'm very happy to contribute to this consortium. So I'm also the PI of a lab that's involved in cancer immunology and immunotherapy of the lung. But obviously we have a lot of experience with immunological assays and we'd like to bring this experience to this project. We plan to leverage the access that we have to samples from individuals who have been exposed to tuberculosis and to use these samples to perform a preclinical validation of the potency of the vaccine candidates that will be developed in this consortium.
So hello, my name is Juan Luis Melero. I work as a computational biologist for Omniscope, where we sequence in high depth the adaptive immune system, the receptors of B cells and T cells. And hopefully our role in BAXERNA will be assessing the efficacy of 3 different mRNA vaccines. And also we will be involved in the clinical trial, comparing the effect of a tuberculosis vaccine by assessing the effect on the expansion of the T cells of the vaccine.
Hi, I'm Bart van der Kerkhoven, Jolien Engels, Louise de Lamane. So we are the GMP team of the University Hospital here in Ghent. We make medicines for treating patients or vaccines for treating patients, and that's why we're part of the BAXERNA team. So once the researchers have developed the vaccine, we will produce it in a way so that it can be used for patients or for healthy individuals.
My name is Isabelle Roux. I'm the head of department of the Center for Vaccinology at the Ghent University Hospital in Belgium. By the time we come into play in the BAXERNA project, the most promising candidate vaccine against tuberculosis will have been selected, and we will bring it from the preclinical to the clinical phase. So we will evaluate the vaccine in a phase 1 clinical trial in a group of healthy volunteers. We will look if the vaccine is safe and how the immune system responds to it. We really look forward to this exciting collaboration.